This is the final test. Can you hear us? We are trying to figure out the sound. Yes, okay, good. Thank you. Sorry about this. Okay. Thank you. Bye. Hello, everyone. Thank you for joining us today for this very first webinar. As you can see, it's always a bit tricky, but we try to do our best. My name is Bastien Kotz, and I'm Communication Manager for Fluigent. Today, Marine Verusel, Life Science Product Manager, will be presenting Automated Cellular Studies with Arya, a new webinar. Just a few notes before we get started. As you can see, you can ask questions in the panel, in the chat. For those who don't have it, it's accessible from the button on the top right of the screen. Questions will be answered by the people on the chat during the presentation, but we also have a session at the end of the presentation for the questions. A recorded version of the webinar will be available from tomorrow. The presentation link will be sent to you. And you can also download the datasheet and the application note from this presentation. Due to the current situation, the webinar is recorded from home, as you may see, and we hope not to encounter any more technical difficulties. However, if it's the case, we apologize in advance and don't hesitate giving us information through the chat. It seems like everyone is here, so it's time to start. Marine, I give you the lead.
Thanks a lot, Bastien. So I'm taking over from here, so I'm going to share my screen with you so that we can access the presentation. Okay. Sorry, I have also difficulties with my computer. It seems I cannot open the presentation. So just 2 minutes. I'm trying things over. And okay, so here we go. So it is my pleasure to welcome you on this webinar on Arya, the new perfusion system from Fluigent. In this presentation, I will start with some context on why we've developed Arya. I will then present how Arya can save you time by automating injection protocol and spare you the mental workload associated to operational work. I will then conclude with an application that we've made at Institut Curie to capture and characterize circulating tumor cells using Arya. So today, hosting you on this webinar was first Bastien, and so there is also me, Marine Verusel, product manager at Fluigent, and 2 R&D managers, William Cesar, who will answer questions related to hardware, and Mathieu Steiner, who will answer questions related to Arya software. So to give you a little bit of context, the idea of Arya came from visiting our clients. So our clients are mainly researchers. And while we were visiting them in their labs, we've realized how busy they are. As a matter of fact, researchers carry out a lot of tasks other than research. So they have all these tasks like analyzing data, plan and perform experiments, review state-of-the-art, and write articles that are mainly related to science. But they also do some lab management, teaching and training, student supervision. They have a lot of administrative tasks to handle. They also apply to grants to have money to have their lab running. And they also attend to a lot of meetings and conferences. So it's a lot of tasks. And actually, to put this into numbers, some studies estimated the time researchers dedicate to each task type. So here's a nice diagram of the time partition of researchers. So you can see that 15% of their time is dedicated to writing articles, but also administrative tasks, teaching. 10% of their time is dedicated to writing grant and applications, 5% to manage data, and that leaves only 40% of their time to actually do research. So it's not much. So to achieve this amount of work during the day, they often multitask. And this is especially true when they perform protocols with multiple injection. They would start office work, and then they would stop multiple times to perform their experiment. So immunolabeling is a good example of a protocol that requires multiple injection. So between fixation, permeabilization, washing, and labeling, there are around 7 to 10 steps to perform. Each step has an incubation time of 30 to 1 hour, so the whole experiment would last approximately a day. And each step has only 10 minutes manipulation. So in total, it's around only 1 hour and a half of effective manipulation. But actually, this is only the theoretical time, because an important thing to notice about interruption, and I'm sure you've already experienced it when you're like highly focused and your phone starts vibrating, like it takes really time to get back to the original task. And some studies have estimated that actually this time is around 25 minutes, so it's quite long. So the true length of interruption would be actually the length of interruption plus this 23 minutes that it takes to refocus to do your task. So knowing that, if we now implement these 25 minutes in protocols with 1 hour incubation, we would have this 10 minutes to manipulate, then 25 minutes to refocus, and that leaves only 35 minutes of effective work. And really, when we take like this 30-minute incubation, it leaves only 5 minutes to really focus. So not much. So if we now go back to immunolabeling and we have 1 hour 10 of manipulation, if we want to add the cost of interruption, we have to add 7 times the 23 minutes to refocus. So actually 2 hours and 40 minutes in total to refocus. So that gives a total of 4 hours of total interruption. So actually 3 times more than the scheduled time. In addition to this, interrupted work also has a personal cost, since it has been observed that people working in interrupted conditions experience a higher workload, more stress, higher frustration, and more time pressure and effort. So now, having this interruption cost in mind and going back to the original partition task in the day of a researcher, if we consider a 9-hour working day, and we know that 40% of their time is dedicated to research, that leaves 3 hours and 35 minutes dedicated to research. Now we will assume that our researcher will perform immunostaining, so meaning 1 hour and 10 of manipulation and in theory, and 2 hours and 25 minutes for office work. But if we implement the cost of interruption, we have 1 hour and 10 of manipulation, 2 hours and 40 to refocus. So that's a total of 3 hours and 50 minutes. And it's actually more than the time dedicated to research. So all this time will be cannibalized by manipulation, and actually manipulation will also overrun the other tasks. So one can conclude that there is a serious need to automate lab work for researchers to focus on science. And actually, there are already solutions available on the market, which are pipetting robots and microfluidics. Pipetting robots are standards, easy to use, and they also look familiar as they look at traditional pipettes. But they require money because they are expensive. They require space because they have a high footprint. They are also dedicated to standard protocol because the only thing they do is they aspirate or withdraw liquid. And they are also limited because they only match with containers like well-plate formats. They also are designed for high throughput experiments, so meaning that if you have medium or low throughput experiments, they won't be profitable for you. On the other end, we have microfluidics. So microfluidic is really precise and versatile as it allows to dispense few microliter at given time points. But it also requires money because it can be quite expensive and long if you want to purchase each single element from different companies to build up your own setup. It requires efforts to acquire this microfluidic expertise, time to build up your own setup, and finally, dedication, because once you have acquired this microfluidic expertise, you want to retain the knowledge and you have to train your colleagues so that they can use this microfluidic setup for their experiments. So we see that the ideal system would be like a combination of the two, a system that would combine the precision and versatility of microfluidic together with the automation and ease of use of pipetting robots. And to address these specific needs, Fluigent has created Arya. And actually, I will run a short video that will present you Arya and its working principle.
Introducing Arya, our perfusion automation tool. Arya makes it possible to automate delivery of up to 10 different solutions in a petri dish or microfluidic chip by following user-defined protocols. Arya's precision makes it possible to inject defined volumes at specific times and to provide time-based sequential perfusion of multiple fluids under controlled flow conditions. Arya's intelligent software allows you to design the protocol that best fits your experimental model. Reducing manual sample handling and procedural complexity is key to improving precision, increasing reliability, and saving time. Even the cleaning is automated. Arya perfuses successive pulseless flows of different solutions, avoiding the extensive mechanical stress associated with classical pipetting. Arya automates protocols such as immunostaining by programming the time delivery of primary and secondary antibodies, the injection volume and flow rate, as well as the incubation time. Arya is the versatile ally you never had. You do the science, Arya does the rest. Introducing.
So now I would like to review with you the 5 benefits of Arya, and I will then explain each of them in more details. So first, the gain in precision, which is quite huge because actually Arya has the precision of the pipette, but Arya also controls the velocity of injection. And we will see in the next slide that it's really an important parameter to control since it can really damage your sample. And Arya is also highly easy to use and intuitive so that you can really focus on science. With Arya, you can perform standards, but also custom protocols, so you can really unlock your creativity and do any protocol you want. Finally, thanks to automation, you can really reduce errors and save time. So first, precision. Manual pipetting is highly precise in terms of volume dispensed, but the velocity at which liquid is aspirated or flushed is not controlled and can seriously damage the biological sample. So if we think about injecting 1 ml, like sometimes we do it in like 1 or 2 seconds, so we can imagine that the flow rate associated actually is quite huge. And depending on users, the flow rate can be abrupt and turbulent. On the opposite, with Arya, the biological sample is really preserved since the velocity of injection is controlled and we are in laminar flow, meaning that the biological sample is not hurt. Another important point is the geometry at injection tip. We don't pay attention to these details, but with conic shape, samples are exposed to important shear stress and unhomogeneous fluid velocity. Again, this doesn't happen with Arya because it has a straight shape at the end, so there is no modification at the injection tip. So actually, for you to visualize the importance of the geometry, here are 2 simulations that show the distribution of the velocity dispersion and the shear strain at the tip wall. So here you see the velocity. In blue would be low velocity, and in red, high velocity. And the same for shear strain. You would have in blue low shear strain, and in red, high shear strain. So you see that really, since the tip has a conic shape, here at the extremity of the tip, you have a really high velocity, and actually, I say high because there is an order of magnitude between these 2 velocities. And here it's the same. We have really an order of magnitude between the shear strain at the bottom of the tip and at the top of the tip. So really, please pay attention of this when you're using your pipette, because you can really hurt your sample. So because of also manual manipulations, there are also a lot of variability that are associated with pipetting. So if I perform the same experiment several times, I have a 5% variability between my experiments. And if somebody one day takes over and performs the same experiment for me, we would have 10% variability between our results. Pipetting also comes with manipulation risk, because when you are manipulating your sample, you have a risk to touch the container sites, so you have a risk of contamination. You have also the risk of using the wrong pipette tip, so not having the right wettability can lead to under-delivery of volume, but also pipetting with an angle can lead to under-delivery. And finally, if you wipe your tips between injection, you can lose some sample. So using Arya, you really completely erase this pipetting risk because you won't touch your sample between injections. And also, since there won't be any manual manipulations, the variability greatly decreases to 0.5% between experiments. So that's a huge decrease in variability, and it really makes every experiment count because you don't lose any experiment along the way. Arya is also easy to use and does not require any expertise in microfluidic. Actually, you can really set your experiment in 3 easy steps. So first, you would load your sample into Arya. Then you would select the solution you want to inject. And finally, set injection speed and volume, and you're good to go. The software have been also designed to be really easy and intuitive, so that you can really generate your protocol as you would write them in your laboratory notebook. So as an example of immunostaining, if I want to perform the following protocol, so first I would like to fix my cells, so I would inject 200 microliter of paraformaldehyde. Then I would incubate this paraformaldehyde for 30 minutes, then wash with PBS for 10 minutes, and finally inject 200 microliter of my primary antibody. So if I want to generate this protocol with Arya software, I will first add a step where I select the paraformaldehyde and select 200 microliter, because this is the volume I want to inject. Then to incubate, I would select the wait function and enter 30 minutes, because it's the time I want to incubate my solution. Then I would inject and wash with PBS. So I select time injection because I want to wash for 10 minutes, and I enter the time, 10 minutes. And finally, I select volume injection because I want to inject my primary antibody, and I want to inject 200 microliter of this primary antibody. So you can see as easy it is to really generate protocols using Arya software. And actually, you can really combine this function to design any protocol you want. So this is really what I meant when I said you can really unlock your creativity, because you really can create original protocols. So you can have like smooth gradient perfusion, diffusion-free injections, long-term perfusion, last-minute sample loading. And finally, an important thing to notice is that you can synchronize Arya with external system. For example, if you do some microscopy, you can ask your microscope to send a signal to Arya so that you can synchronize acquisition with perfusion. So one and the final main benefit of Arya, and this is what my talk is really about, is that Arya can save you time. So if we come back to immunolabeling, we said that we had 7 steps of 10 minutes, so 1 hour 10 of manipulation, but we know that we have a cost of interruption of 33 minutes for each step, so a total of 2 hours and 40 minutes to refocus, so a total of around 4 hours of manipulation. If we now want to use Arya, we will have only 30 minutes to set the experiment, because we would like load all our solution on the system, plug the sample to Arya, and also launch our protocol in Arya software. So let's say it takes 30 minutes. And since we have only one manipulation step, we have only one time, the cost of interruption. So it's a total of only 1 hour. So actually it's a 3-hour gain, which is quite huge. So now I will go into the application to show you what we've done at Institut Curie. So we capture and characterize tumoral cells using Arya. And it was in the team of Stéphanie Descroix. And I really want to thank her, because she is always keen on testing our new products. And actually, it is really what Fluigent is about, like to be close to our customer and to have directly their feedbacks. So, to give you a little bit of context, in aggressive metastatic cancer, cancer cells from the primary tumor travel through the blood system and form new secondary tumors in other parts of the body. So actually, the detection of circulating tumor cells is a good indicator that the patient has a metastatic cancer. But the number of circulating tumor cells is also quite important, because if you number less than 5 circulating tumor cells in 7.5 ml of blood, then the probability of survival of the patient is really high, compared to whereas there were more than 5 CTC, then the probability drastically decreases. It's also important because it will decide which treatment will be given to the patient, because if there are less than 5 CTC, the patient will have hormonal therapy, and if there are more than 5 circulating tumor cells, he will go to chemotherapy. But actually, the main challenge of detecting and numerating these circulating tumor cells is that they are really low compared to the number of the other cell types. So in 1 ml of blood, we will have 1 to 100 circulating tumor cells, when there are like 5 billion of red blood cells, 0.2 billion of platelets, and 7 million of white blood cells. So actually, CTC are really rare biomarkers. But the good news is that microfluidic is actually a powerful tool to detect rare events, due to its great sensitivity. And that's why in the team of Stéphanie Descroix, they developed this chip to capture and detect circulating tumor cells. So the chip is made of 4 chambers, and in these chambers, you have a network of magnetic bits. So if you take a transversal cut of this chip, you will see like you have these columns of magnetic bits, and the magnetic bits are covered with EpCAM antibody because circulating tumor cells are known to express EpCAM antigen on their surface. So the blood from the patient will flow inside the chip, and once circulating tumor cells encounter a magnetic bit, it will be trapped by immunoextraction. So actually, if you want to know more about this technique, please refer to this article. So once the the circulating tumor cells are captured, they can either be labeled, or some molecular analysis like DNA hybridization can be performed directly in the chip, or the cell can be retrieved for further analysis. So this is a multi-step protocols, and the multiple injections were automating using Arya. So actually the steps that were automated were surface treatment of the chip, injection and incubation. So then magnetic bit injections, cell injection, fixation with paraformaldehyde, and finally immunostaining. So the total duration of the experiment was 7 hours, but performing it without Arya leads to a 3 hours manipulation time, and by using Arya, there is a contraction of 1/3 of this manipulation time because it took only 1 hour to perform the experiment. So here are the results of this experiment. So you can see the cancer cell trapped on the magnetic bits, and you can see that we succeed in labeling actin in orange and the nucleus in blue and the DNA damaged in green. So if you want to know more about this application, it's actually available on the website, and I will also put it on the platform so that you can download it directly from the webinar platform. So to conclude my talk, I would say that considering the busy agenda of researchers, Fluigent has created Arya to automate operational lab work for them to focus on high-value tasks. Here are the main take-home message that from Arya. So Arya actually saves you time. It allows you to create standard and original protocol. It reduces errors and improves precision, and that makes every experiment valuable. Finally, thanks to the software, you can share protocols with partners worldwide. If you want to have more detail or a quotation for Arya, please send an email to contact@fluigent.com. I thank you very much for your attention, and now the session is open for question and discussion. So please, if you have any question regarding the talk, feel free to write them in the So is it possible? So I have a question about the flow unit. Is it possible? is it possible to use more than one flow unit? So I will give this question to William.
Okay, so about the question if it is possible to have more than one flow unit. Arya is planned to have a single one flow unit, which can address a flow rate up to 1 milliliter per minute and down to 14 microliter per minute.
So about the minimal volume you can inject with Arya, I think that the minimal volume you can really inject is around 20 microliter. So after how long does it take to purge the system? Actually, like you can do it like really fast if you are in the manual mode, because you can like control only the pressure, and it can be purged in like a few minutes. So thank you, Marine, for the nice presentation. Thank you, it's really nice. Question about TTL. So yes, you can, like Arya has a TTL, so it can send TTL, but it can also receive TTL. So that's the way you can use it with a microscope. So is it possible to introduce air gaps between sequentially delivered fluid to prevent mixing? So I will give this answer to William. So William, can you answer?
Okay, maybe you hear me now.
Yes.
Okay, about the air gap, it is possible technically to inject air gap if you don't fill a reservoir, so you let it with air only. But it may introduce some errors in the volume calculation, so it is not recommended.
So about recirculation, do you want to answer, William? So the question is, can you perform recirculation of fluid in microfluidic chamber with the Arya?
Yes, so this function is not implemented in Arya, so technically you can only inject so far.
So yes, you have to couple it with another system if you want to implement recirculation. So some people ask me to repeat some stuff. So
yeah, maybe I'm not sure everybody heard me correctly. So if you want me to repeat, can you your question so that?
So there is a question of what happens if the outlet tubing is clogged. William, can you answer this question?
If it's clogged, then well, nothing happens really. You have no flow rate, so you will be told by the software that there is no flow rate. But yeah, basically we have a warning, but nothing will happen in your chip.
So after the question is, how do you sterilize the tubing between experiments? Is it just by flushing with ethanol? So actually, like, you can change all the tubing inside Arya. So if you want really to work under sterile condition, you can change them each time. But otherwise, yes, we recommend that you flush some ethanol at the end of each of your experiments so that you really keep it clean. So so the question is, what is the accuracy on the injection volume and how do you avoid cross-contamination between samples? William, do you want to answer this one?
Accuracy is below 50%, above 30 microliters. Well, volume is calculated from flow rate, so we are dependent on the accuracy of the flow meter, the flow unit. And about the cross-contamination, so far there is no solution for now. It would be possible to inject some oily, a small drop of oil or air, but it's not recommended yet. We are thinking about how to improve this.
And also to avoid diffusion between two solutions, we have implemented a clear tubing function so that you can inject your first solution and then completely clear the tubing and inject your second solution. So if you want more details about this, I invite you to leave us your email so that we can have a discussion together and really explain you and go into detail about how Arya is working. Just as a reminder, so now are available the datasheet and the application note I was referring to in my talk. They are available for you to download, so feel free to do. So do you have any more questions? So there is a question about the purge, like how do you prime the system before running an experiment? Because you're right, the system is first filled with air. So the idea is to really get rid of all the air that is inside the system before you launch your experiment. So actually, all these steps are automated by Arya because there is a priming step and it's completely automated so that you prime the system and you don't introduce any air bubble. And again, if you want more details about this, I suggest you leave us your email so that we can recontact you. So to download the files, maybe Bastien can explain you where you can download the files. Bastien, can you tell us where we can download the files on the page?
Hear me? Yes. So the download button is just under the the chat button on the top right corner of the screen. You should have the chat panel, one for polls and one for download documents.
So is it okay? Did you find the files? So there are other questions about the minimum and maximum flow rate with Arya. There is no document in the folder.
If the documents are not available, we'll try to send them by email after the presentation.
Yeah, it's weird because
sorry about it.
So there is a question about the minimum and maximum flow rate, and is the flow rate directly controlled from the software? So I leave this question for William.
The minimum flow rate is 40 microliter per minute. You can go below, but then you will have not very good accuracy. And you can go up to 1 milliliter per minute. This depends also on the resistance of your chip, because if it is really resistive, we cannot pressurize enough to ensure that you will reach 1 milliliter per minute. But with main chip used for this application, in general, they are not very resistive, so it's okay to go up to 1 milliliter per minute. And about the flow control, it is control locally on the machine, but the software have an eye on that, so it will display the volume directly.
So we have another question about the dead volume between sample aspiration and fluid outlets.
Internal volume is about 200 microliter. Then it depends on the length of the tubing you place between the two-switch valve and your chip. The two-switch valve is used right before you chip to either select your chip or waste reservoir, so that you can clean the machine and perform the prefill. So it is the mandatory part of Arya. And then you are free to use the length of tubing you want, but we recommend to use it as small as possible so that you reduce the total volume.
So another question is, can it run without a computer? So actually, yes. So it's really built up so that you can use it either manually or automate your protocols with with the software.
So we'll wait a few minutes in case we have a few more questions, and then we'll close the webinar. Okay, I don't see any more questions. Oh, yes, obviously one. Marina, let you answer it.
Do you have? I don't really understand the question.
Do you have to tell how long is your tubing for injection of a previous small sample? Do you mean that when you prime the instruments, it calculates automatically the length of the tube?
Yes,
If that's
there
the
is
question.
yes, there is a calibration step so that it automatically calculate for you the volume between the reservoir and your sample. Can you automate without a computer directly on the screen? Unfortunately, no. I'm sorry. So you will need a computer to automate your protocol. Then something is wrong with me. Yes, we can definitely share the files at the end of the meeting. No problem. Can you put Arya inside an incubator for cell culture? So actually, you cannot put Arya inside your incubator, but what you can do is put Arya close by your incubator and have enough tubing so that it warms up the tubes before it enters your sample. And how does the PC connect to the Arya? So yes, it is USB. Can you chain multiple units together and operate sequentially? I'm not sure I understand the question. William, do you want to answer this question?
So you could imagine having a kind of network of area connected to a single ship, but they could not be synchronized. So at the moment, it is not possible to make them communicate except from TTL. But in terms of software, it is not possible. But you can imagine having several areas connected to a single chip.
So after there is like a really specific questions, and so Sylvain, can you please send me your email so that we can recontact you and discuss more in details with you actually what you want to do, so that we can really address you the fluidic system that would better fit your request. So thank you, Leslie, for your question. So are there other systems offering a similar spec? And do you organize workshop where we can try Arya with our sample? So actually, Leslie, if you can send us also your email, then I will have our sales contact to contact you so that you can really try Arya directly in your lab. I think would be the best way for you to see how it runs. And to my knowledge, there is no system on the market offering similar spec. Thank you, Max, for your nice message. Thank you, Charlotte.
We'll wait a few minutes if we have more questions.
Thank you everyone for attending. It was really nice discussing with you, and thanks a lot for all your questions. It was really interactive, so I really appreciated it. And sorry for all the sound and the image at the beginning, but I think it went pretty well in the end. Thanks a lot to all of you.
Well, I think it's time to close the webinar. Thank you, Marine. Thank you, William, for answering the question. As a reminder, if you have any question, feel free to contact us at contact@fluigent.com. You will receive shortly an email with the recording of the presentation. We try to also send you a recap of the questions and the documents that were attached. Have a look at our website for more webinar. We have a specific page for the upcoming webinars. And after that, we wish you a pleasant rest of your day. Thank you for attending. Bye, everyone.
Bye.